Hello, everybody. Welcome to yet another talk, another episode of our Western Biology Evolution Seminar Series. Just as a quick reminder, this is an online series of talks based or focused in evolution. So today, here I have Isabeau Lewis. She just finished her undergrad at Queen's, and she will present the work she has done during her undergrad. And just a little bit of a background information. I've known Isabeau because she attended a conference we organized earlier this year, the OE3C, Ontario Ecology, Ethology, and Evolution Colloquium. And she presented her talk there and she won an award. So I'm more than sure the talk today will be great. So Isabeau, please take it away.
Thank you so much, Mateus, for the introduction, and thank you for inviting me and having me here. I'm so excited to tell you about the work I did on plant responses to dynamic environments during my undergrad. Like Mateus said, my name is Isabeau and I'm from Queens. So just so you know what's to come, we'll start out with a quick introduction to sessile plants in variable environments. Then I'll spend about 15 minutes telling you about plant responses to neighbor identity, focusing on an independent project I did looking at kin discrimination in plants. After that, for about 20 minutes, we'll move on to plant interactions with pollinators, which was the focus of my honors thesis research. And finally, we'll end off with a quick conclusion to wrap everything up. So let's go. So just to start, I'll take you back to my title slide. This is a picture I took of my field site, which was handily a literal field, and we have some lovely milkweed right in the foreground here. I'd like you to picture a plant in this field over the course of its life, from seed to seedling to vegetative growth to flowering and reproduction. Now picture all the other plants in the field alongside it, also growing and changing the landscape, nutrient availability, and number of potential mates. Add in changing environmental conditions and pollinator visitation, both within and between years, and this is a lot for an individual to deal with. Because a plant can't predict where its seed will land or go find more favorable conditions, plasticity can be crucial to dealing with spatial and temporal variation. It's the extent of this plasticity that really fascinates me and what my work so far has focused on. So one factor that I've alluded to affecting these plant responses is the presence and identity of neighboring plants. When plants are surrounded by close neighbors, they compete with the rest of the patch for the same amount of limited resources. Because of this, plastic responses that increase competitive measures in the presence of neighbors can also increase fitness of individuals. So in this example, Franklin and Whitelam tested responses to competition for light by growing plants in different conditions, simulating shading or no shading by neighbors. During both initial vegetative growth, which is on the left, and flowering, which is on the right, plants that were shaded responded by elongating their stems, which would increase access to light if the shading was actually coming from taller neighboring plants. But competition between neighbors isn't always so straightforward. Because of things like clonal growth, self-fertilization, and the limited dispersal of pollen and seeds, plants within a patch frequently neighbor their own relatives. In these cases, the outcomes of competition with neighbors depend not only on the resulting direct fitness of the individual, but also of the consequences of competition on its related neighbors. In general, if this decrease to indirect fitness is costly enough, altruistic behavior towards relatives can evolve, provided that the fitness benefit to the relative offsets the cost to the individual. Because this requires the capacity to recognize the identity of neighbors, for a long time, plants were not thought to not exhibit this kind of social behavior. But plants do have the ability to recognize the identity of others, for example, through self-incompatibility during fertilization, the recognition of clonal growth, and through root exudates secreted through the soil. And of course, as we've covered, plants also have the capacity to respond plastically to their neighbors, setting the stage for kin discrimination in plants. Although studies of kin discrimination in plants are still pretty rare, there are examples of both vegetative and floral responses to kin. For example, Dudley and File examined below-ground root responses to the presence of kin. They grew plants of Cachyolidentula in pots where all were half-siblings, all unrelated to each other, or grown alone as a control. They then weighed both the root and the shoot mass of the entire pot. In the graph on the right, the x-axis shows the presence or absence of neighbors, with kin in black and non-kin in white. The y-axis shows the proportion of total mass allocated to roots. As you can see, non-kin pots allocated more to their roots than did their kin counterparts. Increasing roots can increase nutrient uptake, so in the presence of non-kin, this may allow plants to draw more resources and outcompete their neighbors, where they stand to gain no benefit from their neighbors getting resources. Then on the above ground side of things, Teresa et al. Looked at flower responses to the presence of kin. They grew focal plants in the middle of the pot and surrounded them by kin or non-kin neighbors, and then replicated this for 2 different density levels, so either 3 or 6 neighbors. They then measured the floral display advertising effort of the focal plant in each pot. For the graph on your right, the number of neighbors is on the x-axis, and the floral advertising effort is on the y. Kin-neighbored focal plants are shown in orange, and those neighbored by non-kin are shown in blue. At the 6 neighbor density, focal plants neighbored by kin invest significantly more in flowering than do ones neighbored by non-kin. Because flowering is costly to produce, this may be a cooperative effort on the part of kin neighbors to increase pollinator visitation towards relatives. So although flowers are expensive to produce, higher flowering can attract more pollinators to a patch. Increasing flowering when neighbored by kin can then increase both visitation to the focal plant and to its relatives, thereby increasing inclusive fitness. However, there are no equivalent benefits to pollinators visiting unrelated flowers in a patch, so flowering less with non-kin neighbors can avoid expending valuable resources. However, while these are some examples providing the basis for kin discrimination in plants, the literature is still really limited and many questions remain. For example, do both floral and clonal growth respond simultaneously to the presence of kin? So this is where I come in. As an independent research project in the 3rd year of my undergrad, my supervisor, Dr. Janice Friedman, and I were really interested in looking at floral and clonal responses to kin at the same time. For this, we chose Mimulus guttatus, which is this beautiful plant on your left, because it reproduces both sexually with these yellow flowers and clonally with these clonal offshoots called stolons that you can see running along the bottom of the pot. So for this experiment, I predicted that focal plants neighbored by kin would increase floral display size because there are higher rewards for attracting pollinators to a related patch. Conversely, I predicted that clonal and vegetative growth would decrease to reduce resource competition and crowding between relatives. And then for non-kin neighbors, I predicted that focal plants would decrease floral display size, but increase vegetative and clonal investment to maximize their use of available resources in the patch. So to actually test these predictions, I grew 1 focal plant in each pot and surrounded it by 4 neighbors. As kin, I surrounded a focal plant by either 4 self-sibling neighbors from the same family for a relatedness greater than 0.5, or outcross siblings from the same family for a relatedness roughly equal to 0.5. Finally, I surrounded focal plants by 4 non-kin neighbors, each from different families. I then grew them in the greenhouse for 9 weeks and measured floral and clonal investment for a total of 225 focal plants. Yes, that took a lot of measurement and time, but it was worth it. So here are the plants from week 1 to week 6. They got even bigger than this by week 9. This was my first time ever having experimental plants of my own, and it's a terrifying leap of faith to watch these tiny little seedlings and just hope that they grow, but it's so rewarding when they do. And also a special shout out here to my supervisor, Dr. Janice Friedman. Because we had to create families of plants for our treatments, we had to cross-pollinate plants and carefully track their lineages. Crossing can be a super delicate affair, and the timing to it is really specific. You have to get it all done during the window when the plants are actually flowering. Unfortunately, the window for this experiment's pollination happened to be right at the time when our entire lab came down with COVID. So this is Janice at home pollinating plants while in isolation and sick with COVID. That is some dedication. So now I'll move on to the results of this. I found that plants significantly increased growth performance in the presence of non-kin. So on the x-axis for both of these graphs are the selfed, outcrossed, and non-kin treatments. And on the y-axis are measures of flowering and of clonal growth. For both flowering and stolons, focal plants invested more when neighbored by non-kin. We also see a trend where effects are pronounced in the self-treatment, which is in the direction of relatedness. By increasing stolons, focal individuals neighbored by non-kin may crowd out their neighbors and access more resources. However, opposite to what I predicted, floral display investment also increased in the presence of non-kin. This may be because of competition rather than cooperation for pollinator attraction. When plants are neighbored by kin, they gain fitness from pollinator visits to relatives. Because of this, pressure to attract pollinators to the individual over other plants in the patch decreases. On the other hand, though, competition to have pollinators visit a focal plant over its neighbors increases when those neighbors are unrelated. Larger displays can increase the chance of a pollinator visiting a plant, which may allow a focal plant to behave more competitively in the presence of non-kin. But we also need to consider these focal plant responses relative to the behavior of their pots. Here, the x-axes represent the growth, or generally productivity, of neighbors in the pot, which is either the number of flowers in the pot or the total biomass of the neighbors. The y-axes are measures of floral display and clonal growth, with each treatment represented by a different color. A steeper slope will tell us that plants are investing more in growth relative to the productivity of their pot. First, looking at the non-kin pots, which are in orange, they show the greatest change in both floral display and clonal growth as productivity and consequently competition increases. If the predominant social interaction between plants is competition, plants may benefit from reducing competition amongst kin and increasing it with non-kin. This is especially true in high productivity patches, where competition for pollinators and resources is the highest. And indeed, we see no comparable response by plants neighbored by kin. So you can see that in the blue and the green, which represent the self and the outcrossed respectively, as the productivity of the pot increases, there is no competitive response in the productivity of the individual. So do plants respond both florally and clonally simultaneously to the presence of their neighbors? Yes, indeed they do. Focal individuals of Mimulus guttatus increase both floral and clonal growth in response to the presence of kin, which indicates a role of competition in facilitating these social interactions between plants. Co-plasticity. Now, I've been talking kind of about whole plant plasticity over a broader growing season or life cycle, but plasticity can also occur on finer scales, including within individual flowers, which is what I'll be talking about next. So flowers all share a single goal, which is to reproduce, but are incredibly diverse. I'm interested in understanding the causes and the consequences of this reproductive diversity. For animal-pollinated species, one crucial driver of diversity is selection by pollinators. For example, it's important to both attract pollinators and to attract the right pollinators. These two phlox species, when growing allopatrically, both have these purplish flowers, but curiously, when they overlap, Phlox drumondii has a higher occurrence of this red color morph, whereas Phlox cuspidata stays the same light purple. Hopkins et al. Found that this was due to reinforcement between the two species. The graph on the right shows the relative fitness of several different Phlox drumondii color morphs in allopatric and in sympatric ranges. When no Phlox cuspidata is present, all morphs have similar fitness, and in fact the light purple is the highest fitness. But when the two species grow together, the red drumondii morph that's most distinct from cuspidata has the highest fitness. This is because hybrids between the species have high sterility, so selection to have distinguishable flower color is strong to avoid pollinators facilitating cross-pollination between the two species. But there are other factors complicating successful pollination. Many flowers are hermaphroditic, meaning they share male organs called anthers and female organs called stigmas together in the same flower. Hermaphroditic flowers have to balance three functions then: pollen donation, pollen receipt, and advertisement to pollinators. While being hermaphroditic can allow flowers to decrease the costs of showy petals, which are expensive to make, because they're shared between the sexes, it can also introduce conflict or interference between them. For example, if anthers and stigmas have the same ideal position for these pollinator visits, interference between the two can actually reduce overall fitness. When these fitness consequences are high, including for both interference and selfing, we see hermaphroditic flowers evolving additional strategies to limit contact between anthers and stigmas. Flowers might have a spatial separation between the two sexes. Or they might have a temporal separation, which is the strategy I'll be focusing on. In this flower, male function comes before female function. Once male anthers have released their pollen, they move away so the stigma can come up and receive pollen in the same position. Just a technical point here that this happens in the single flower. I've shown it here with two so you can see what both male and female phase look like. What's especially interesting about this temporal separation is that male and female phases have different limitations to fitness. Male fitness generally depends on fertilization success, whereas female fitness is limited by resource investment. So the benefit to the amount of time allocated to each sex function and the benefit to pollinators visiting flowers of each phase may not be the same. This may lead flowers to evolve other signals of sex phase for pollinators. This brings us to Saponaria officinalis, which is my lovely study species for this experiment, despite it being an invasive weed that's kind of everywhere in Ontario. Saponaria, as I'll refer to it now, has a temporal separation between anthers and stigmas, and also changes color of its petals as it changes sex phase. So flowers start out white and in male phase with exposed anthers that release pollen. Then they go into female phase, and with that, their petals turn pink. As they transition into female phase, the anthers shift out of position or fall off, and the stigma comes up to receive pollen. They don't always turn the same shade of pink, though. Here's a photo I took of two female phase flowers from the same plant. They're both at the endpoint of female stage, but are really different colors. I hypothesized that this might have occurred if their sex phase and color transition had taken place at different rates. For instance, if 2 days after opening, the bottom flower had still been in an earlier phase. What's more is that pollinators discriminate against the flowers they visit based on these colors. So these are findings from a paper by Davis et al., who created floral displays made up of either all white male phase flowers, all pale pink female phase flowers, or all vivid pink female phase flowers. They looked at how many flowers pollinators visited on each type of floral display. A higher bar means that pollinators visited more flowers. You can clearly see that pollinators visit more white male phase flowers compared to either shade of pink. Male fitness depends on the amount of pollen removed, and every pollinator visit increases that linearly. On the other hand, female function relies on fertilizing and nourishing potentially just a few large ovules. So the pollen needed to fertilize them can be brought with just a few pollinator visits, and after that, female fitness from pollinator visits saturates. So this color change may be an adaptive strategy to manipulate pollinators to visit more male phase flowers, where increasing pollinator visits will have greater impacts on fitness. But there aren't always the same amount of pollinators around, which can change the benefits to investing in time in male function, which is removing pollen, versus female function, which is both receiving pollen and setting seed. And as you have seen, flowers don't always invest in sex function or in flower color transition in the same way. What I wanted to focus on for this project was whether the amount of pollen removal could be causing the variation we see in flower color and in sex investment. So with this, I asked, do flower color and sex phase in Saponaria respond plastically to different levels of pollen removal? And if so, what do these responses actually look like? I predicted that under low pollen removal, such as when there are a few pollinator visits, there would be high remaining male reproductive potential, meaning that increasing pollinator visits would be crucial to maintaining overall fitness. In this case, I predicted that flowers should stay white to attract more pollinators and prolong male phase. Conversely, when, for instance, many pollinators visit and most pollen is removed, remaining male reproductive potential decreases, and fewer pollinator visits are needed to donate all pollen. In this case, I predicted that flowers should accelerate pinkness and female phase and direct pollinators to visit other, more rewarding flowers that have not yet been visited. So to actually test these predictions, I needed to break this main question down into 3 parts. First, does pollen removal affect flower color over time? Second, does pollen removal affect flower color over sex phases? And third, does the timing of sex phase transition itself change with different levels of pollen removal? So in the summer of 2022, I headed out into the lab with Ali Vanato, a PhD candidate from the Friedman Lab, to actually test these predictions. This photo of Ali is from our 2023 field season, so shout out to Candace Ma and Sarah Gamma, who were 2 of our field assistants from that year. In the field, I conducted a common garden experiment at the Queen's University Biological Station, which is just outside of Kingston, Ontario. For this, I manipulated the level of pollen removal, as you'd see under different levels of pollinator abundance. So for my first treatment, I bagged flowers so that no pollinators could visit them and no pollen would be removed. For my second treatment, to simulate high pollen removal, I removed the anthers from flowers. This is because pollinators knock anthers off during their visits, or they fall off after they've donated all of their pollen. I should also point out here that removing the anthers was actually a lot more delicate than this graphic makes it look. I used forceps and I did all the treatments in bud, so I had to really carefully pry open the petals to do this. And then finally, I left flowers unmanipulated and open pollinated, just so I'd have a reference point for how flower color and sex phase typically change. For this overall, I manipulated 450 flowers in the common garden across 50 plants, embedded within a larger experimental garden of 1,600 plants. This is a drone shot of one of our plots, which was so cool to get. Shout out to Alan Tian for making that. And there's me for scale, and I think my field assistant Erin from 2023. So I did all the treatments on every plant, so each one had bagged flowers like in the photo, ones whose anthers had been removed, and open pollinated ones. As I mentioned, I treated flowers when they were in bud to make sure they would all be at the same floral stage. I then measured them at 1, 2, and 3 days after opening so I could get a sense for how flower color and sex phase changed over time. To measure color, I used a UV spectrophotometer, which is this device that you can see here. It gives you these reflectance graphs, which show the amount of reflectance of a visible spectrum of light. I then use this to create a measure of how pink the flowers were. So you can see that pinker flowers are characterized by a reflectance curve with a valley right around here. So they have higher blue and red values, making them pink. Whereas white flowers have a pretty consistent straight curve all the way across, meaning they reflect the same amount of light and are white. So when I measured flower color, I also assigned flowers to one of these phases based on the relative emergence of the stigma to characterize the sex phase transition. So for early male phase, there's no emergence of the stigma, it's just the anthers open and releasing pollen. For late male stage, the anthers have started to dehiss and the stigma has started to emerge, but is kind of not super visible. You can kind of make it out right in the middle there, but it doesn't look super distinguishable from the anthers. And then by early female phase, the anthers have fallen off or at least shifted to the side, and the stigma is starting to emerge from the middle, but isn't yet fully receptive. And then in late female phase, the stigma is fully receptive. It has these curved lobes that you see here, and yeah, can receive pollen. So now I'll move on to my results. For my first question, whether flower color changes over time. To orient you to this graph, on the x-axis is the day of measurement, and on the y-axis is a measure of pinkness using the output of the spectrophotometer. A higher value indicates a pinker flower. The pink line is when all pollen is removed, simulating when many pollinators visit. The black line is open pollinated, and the blue line is when flowers are bagged so that no pollinators can visit. On the first day after opening, you can see that all flowers are pretty white. They've got a low pinkness value. By day 2, flowers are pinker, and by day 3, all the flowers are a lot pinker. But when we compare between treatments, we see that they're not actually responding in the same way. So flowers that have no pollen removed are staying a lot whiter even at 3 days after treatment compared to flowers that have all pollen removed, which are significantly pinker. This is presumably because flowers with all pollen removed have no more male fitness to be gained, so they may only need a few more pollinator visits to fulfill reproductive output. On the other hand, flowers with no pollen removed need many pollinator visits to gain fitness. Then, by staying white, they can attract more pollinators to them and fulfill that reproductive output. So now on to my second question, whether flower color responds differently over sex phases. So to orient you to this graph, on the y-axis is the same measure of pinkness, but this time on the x-axis is the sex phase transition I walked you through earlier, which moves from male to female phase. You can see that in early male phase, flowers are pretty white, and then they gradually increase in pinkness throughout the sex phase transition. But when we look in between treatments, we actually see no differences in pinkness at each sex phase. So what this means is that sex phases have the same pinkness no matter the level of pollen removal. Although I will point out that it does trend in the direction I expected, with flowers at late female phase and all pollen removed being slightly pinker than flowers with no pollen removed. But still, this isn't really in line with what I expected to see. So when we look at whether sex phase transition timing itself changes across treatments, these results become clearer. For this graph, on the first panel is day 1, then day 2, and then day 3. Each day is separated into treatments, ordered by no pollen removed, N, then open pollinated, O, and then all pollen removed, R. On the y-axis is the sex phase frequency, or the proportion of flowers in each sex phase for a given treatment and day. Blue colors represent male phases, and pinks represent female phases. When no pollen is removed, the N section, flowers start out in male phase and are still over 50% in early female phase by day 2, and then finally at day 3 have a majority in late female phase. On the other hand, flowers that have all pollen removed are over 50% in early female phase already by day 1 and reach late female phase faster. This may be adaptive, allowing flowers to adjust the timing of their sex phase transition to their real-time reproductive capacity. But as we saw with my second question, sex and color may be linked, leading to a constraint on plasticity. Or if signal honesty is important, then that may be a useful strategy. So the answer to my initial research question is yes, saponaria flowers respond plastically to different levels of pollen removal, specifically by accelerating the transition to pinkness and to female phase when pollen is removed. But as you may have noticed, I found the most changes in color at day 3 when the majority of flowers are already in female phase. So what about color changes in response to pollen deposition? It's possible that they continue to increase in pinkness throughout female phase, so after day 3. I was so curious about this that the summer after I graduated, so 2023, I headed back out into the field to conduct a follow-up experiment manipulating pollen deposition. This time, because flowers would already be in the last sex phase by the time I manipulated them, I focused on just flower color responses. While many visits may not be required for female fitness to saturate, at least one or a few still are to bring in pollen. So I predicted that if there was no pollen deposition, flowers would stay lighter in color to continue attracting pollinators. Conversely, if a lot of pollen was deposited, I predicted that flowers would turn a darker pink to direct pollinators to other flowers requiring more visits. And with that, I headed back out into the field with Erin Pagulayan, who was my incredible field assistant for the season. These experiments are not an easy feat. For them, you generally have to work every day to track the flowers at each day, and he took it with so much grace. So shout out to him. So this time around, I bagged flowers until the 3rd day after opening because I didn't want there to be any effects of pollen removal or uncontrolled deposition before the flowers entered female phase. Then, at day 3, when flowers were in late female phase with the most receptive stigmas, I conducted 3 different pollen deposition treatments. First, I bagged flowers so that no pollen was deposited. Second, I supplemented pollen to flowers until their stigmas were saturated. This treatment I repeated each morning before measurement to ensure sufficient deposition. And finally, I left flowers open pollinated to see how they would typically change in response to pollen deposition. I then measured responses at 4 and 5 days after opening. I had initially intended to do this until day 6, but generally by day 6, flowers' petals are so wilted that they're unmeasurable. So that's kind of the total floral advertisement effort. Okay, so now I'll move on to my results for this one. I'm showing them here with the pollen removal treatments, so you can compare the pinkness after pollen removal and deposition. So for this graph, on the y-axis is our same measure of pinkness again, and this time on the x-axis is the number of days after opening. So you can see that in 2022, I measured at 1, 2, and 3 days after treatment in response to pollen removal. And then in 2023, I'll show my days 4 and 5 in response to pollen deposition. And all the treatments are listed along here. So adding in my 2023 pollen deposition treatments, you can see that after one day after pollen deposition, so on day 4, flowers are of a comparable pinkness to flowers 2 or 3 days after pollen removal along here. However, by day 5, all flowers are much pinker than they were on the previous day. Particularly, flowers that had supplemental pollen deposited have greatly increased in pinkness over here, but those with open or blocked pollen, so the blue and black lines, are of comparable color to each other and haven't shown an equivalent increase in pinkness. So flowers responded differently in the extent of their color change over time, depending on the level of pollen deposition. So the answer to my follow-up question is also yes. Flowers adjust their floral advertisement in response to pollen deposition. If pollen is limited, then flowers that have not yet had any pollen deposited may gain fitness from remaining attractive to pollinators in female phase. But why maintain flowers that have fulfilled reproduction instead of just letting them wilt faster and allowing more resources to be allocated to other functions? Overall floral display size can increase pollinator visitation and, in turn, reproductive success. So, retaining pollinated flowers may allow plants to create larger displays to attract pollinators from long distances without the resource costs of producing entirely new flowers over and over again. Then, when visiting a plant, a color change can direct pollinators to flowers with higher reproductive potential. An alternative explanation for this color change is that the accumulation of anthocyanin pigments, which cause the color change, may function to protect maturing seed from sun damage. Anthocyanins also accumulate in the calyx, which is where the seeds are over here, and the calyx remains intact after the petals wilt. So anthocyanin accumulation in the petals may occur as a consequence of anthocyanin accumulation in the calyx following pollination to protect the now growing seed. But what I'd really like you to take away from this is that we often think of plant-pollinator interactions as being dynamic, but on the part of the pollinator. What this work shows us is that plants are manipulating pollinators and their environments right back, not just on larger evolutionary timescales, as I showed you at the start, but also within individual plants and even within individual flowers. And more broadly, plants interact with their environments in incredibly complex ways, the extent of which we are still discovering. Things like kin discrimination have really not at all been examined in plants, but they have a lot of potential to influence the relationships between neighbors in a patch, or the relationships between plants and their pollinators, as you saw with my flower color project. I hope that I've been able to inspire you to wonder a little more about what plants are up to, and consider all the fascinating processes occurring in natural environments, even just regular fields. So with that, I'd like to say thank you all for listening. Thanks also to my supervisor, Dr. Janice Friedman, who always goes above and beyond, and everyone else listed here for their invaluable help making these projects possible, particularly Ali, Christina, Claire, Erin, Sarah, and Candace. And thank you so much, Mateus, for having me in Western. And with that, I'll move on to any questions.
Wow. Thank you very much, Isabeau. During the conference, I didn't have a chance to attend your talk. I was running around, but now it comes to me as no surprise why you got the award. And not only you were a great presenter, but also the topic's fascinating. Although, I must say, I mean, kin recognition in plants is not totally alien to me. I've heard about and I've certainly thought about it. I think your experiments really, yeah, they bring this to focus, right? I was very interested when you started talking about it and showing your results and how some of your results actually contradicted your original expectations. I found it fascinating. And like you mentioned, I'm assuming that kin recognition in plants is very, very poorly studied, right?
Yes, yeah. So it hasn't really been done, although I will say I can't remember his name anymore, but at OE3C this year, there was someone who did his master's on kin discrimination. So I think it's hopefully coming up.
I
Yeah,
think
it has, yeah.
it might have been Walbert Wu, I think.
Yes, that's
Yeah,
him.
because I remember his talk.
Yeah, yeah. So there's maybe a movement for more of it, but yeah, definitely still understudied in my opinion.
Hmm, hopefully, yeah. So now, I have a few questions pertaining the first part of your talk, kin recognition in plants. So you touched upon why you found results that were unpredicted. Now my question to you is, do you like how common, how widespread would those phenomena be in a larger ecosystem with different species? What do you think? Like, how do your results translate to the field?
Yeah, so I think it really depends on the species you're studying, which is why more studies of kin discrimination are important because we don't really have a generalizable rule yet. I will say, though, that it also depends on the species because of the growth method. So, for example, as I mentioned, Mimulus guttatus grows clonally, and what that often looks like in the field is large patches of a single species, just Mimulus growing clonally together. So, it's often with conspecific neighbors and specifically relatives. So in circumstances like that, kin discrimination may be more important to helping plants, and that's where we might see the most responses. It can also depend on the level of inbreeding depression. So another potential effect of mating with close relatives or flowering a lot around close relatives
Mhm,
um, is inbreeding depression. So if that presents a high cost in the species, then we may see kin discriminatory responses in that direction. So yeah, it's hard because I can't give like a general rule of thumb yet, and that's why I think we should have more work.
mhm,
But I do expect that in species where they're often interacting with the presence with relatives, or there are consequences to those like interactions with relatives, that we would see similar kin discriminatory responses.
mhm, mhm. Yeah, you stole my thunder with the inbreeding depression problem. I was gonna go there.
Yeah, that's okay. You can still go there.
Uh, no, my second question is more actually, um, I might have not understood one part of your methodology from the second part of your talk. So when you were measuring, the different color patterns in the
Mhm.
flowers, did you care whether those flowers came from the same plant or because what I'm thinking here is what about the cascading effect, when let's say there's a threshold within one particular floristem. Well, I'm not really sure how the flowers are organized in that species, but let's say, if you have like 3 to 4 flowers that have reached a certain level of pinkness, whether they
Mhm.
will, ignite a cascading event on other flowers. Like, were you able to separate that or not? What do you think?
Yeah, that is an excellent question. I'll just flip back to a slide that kind of shows the way it
Hmm.
flowers. So Saponaria produces a lot clonally in addition to flowering. So the plants in my experimental plot had multiple stems emerging from the ground. Um, and so I was able to actually um, do different treatment. So each time I would treat a flower, I would pick a new stem to treat it on. And I also did every treatment on every plant in randomized order. I don't know if that makes sense. So for one plant, for example, it would get um, no pollen removal first, be measured at day 4 and then day 3, and then they'd day 2 for different flowers. And another plant, it would start with open pollination.
Mm-hmm.
So that was how I accounted for potential variation, both in having the same stem flowering and also in terms of like the weather it experienced or the pollinators that happened to be around in that time. I then also included plant ID as a random effect in my models. Yeah, just to account for plant specific differences in color. But
Mm-hmm.
yeah, to go back to the specifics, I tried where I could to do it on different stems, and I never picked flowers that were just exactly adjacent to each other. Luckily, these plants flower a lot, especially in the second year, so I was generally able to find like 4 or 5 good flowers at least a day to measure that were on pretty different parts of the plant.
Mm-hmm. Now,
But yeah,
just
that's a really good question.
thinking about also dynamics of conducting the whole research project. So the flower, the color, the flower color project, you said it took 2 summers, right?
Yeah.
But I'm assuming, so the other project with kin discrimination, that it was in the greenhouse, so you could conduct during winter in a.
Yes. Yeah, that was during my winter semester of third year. So that, yeah, it was a really different vibe to move from the greenhouse to the field when I started my honors thesis. But
Yes, I can imagine. So in
yeah.
total, in the Friedman lab, how long were you there for, roughly? 2 years?
I was there, so I started working as a research assistant the summer after my second year, and I just finished the summer after my fourth year, so like 2 full years total.
Mhm. Yeah,
Yeah.
well, I'm asking you all these questions because I think your talk really illustrates how much we can do, right? When you are, I'm more than sure that the Friedman Lab is a conducive environment to great discussions, and you were well nurtured and well supported, and it was beautiful to see how many results you have. Again, I'm not surprised that you got the award in the conference because the talk was great. You really brought. You started by saying, oh, this is my undergraduate work. And usually I think people are shy to present their undergraduate work, but this is graduate level work. So congratulations. I think it is a great way to illustrate how much we can do when we have the right support, when we have dedication, right? You must have worked really hard for this. So congratulations.
Thank you so much. Yeah.
Yeah, and I also found your talk, the topics and the things you found, what you presented, really speaks to the goal of this seminar series, because my goal when I created the series really was, well, first and foremost, to give speakers a platform for them to promote their studies, of course, but also to show people, whoever watches us, to show how evolution is, multifactorial, right? So multifaceted, in which you can study evolution in so different aspects. And like you mentioned in the very end, and short time spans, but also like, because usually when you think about evolution, you think about millions of years, right? So
Mhm, mhm.
macro evolution. But here really show you were dissecting plasticity, right? In real time. And you really showed the interaction between pollinators and plants. So yeah, this was great. Thank you very much.
Thank you so much. Yeah, I'm so grateful to have this opportunity again. Like as an undergrad, that's not often something that comes up is to give a whole seminar on your work. So yeah,
Yes.
I feel very fortunate to do this. And yeah, I'm eternally grateful to all the grad students in the lab. I think like, you know, going through your lab, just yeah, being kind to the undergrads and
Mhm,
nurturing them is super, super important. And I know I've benefited a lot from it.
mhm,
And then also if there's undergrads watching this, like yeah, don't be afraid to do science. It's so cool. And also I think you're in kind of a unique position as an undergrad because like for my kin discrimination project, for example, it's not like it was a pretty small project and not really something that could be done for an entire master's,
mhm,
especially because it's kind of a new thing and it's not really, certain like you'd want for a master's project. So I think undergrads have really valuable opportunities to
mhm,
to do new science and to do super cool science. So yeah.
mhm. I agree. They can be like, they can take on riskier projects and just
Yeah.
play with it. Yeah, I
Yeah,
agree.
exactly. Yeah. And just, be there to learn and find cool stuff sometimes.
Yes, wonderful. Well, thank you so much, Isabeau. I'm pretty sure your talk will be valuable and inspirational, not only to undergrads, but pretty much to anyone who watches it. So thank you very much for being here with us today.
Thank you so, so much.
So I will stop the recording now, okay?